Hi, everyone. Hi, Renee. And Nina. We're giving a few minutes for others to join, so we're just waiting. Hi, Eric. How are you?
Good afternoon. How are you doing?
I'm well. Yeah, so it's 3 o'clock now. Thank you, everyone, for connecting so punctually, and we'll just give one more minute for everyone else to connect. In the meantime, I'm gonna test the screen sharing so that you can all see the agenda. So 1 second while I put it up there. So can everyone see the slide the good way?
Looks good.
Yeah, perfect. And this is the agenda. So in 30 seconds, we'll start with our intros from Dr. Scan and Nestler, and I'll go back to seeing everyone's faces. So I'll stop sharing my screen. Okay, so it's now 3:01, and you're at our annual virtual research training program, Meet and Greet in the Department of Psychiatry. So this is for applicants interested in our R25 and T32 funded research training programs in residency and fellowship. And we'll start by hearing from Dr. Renee Kant, who's the chair of our department and also a very accomplished physician scientist himself.
All right, thank you very much, Mercedes. Welcome everyone. This is indeed, one of our prominent programs where we try to train people in the combination of clinical work as clinicians and researchers. And I always start saying that I was probably the first in this program many, many years ago when our former CEO, who was at that time chair in psychiatry, we're talking about the 1980s, 1990s, started this program with the goal of training psychiatrists, clinicians as researchers. And like I said, I was one of the first, probably the first, who combined a residency with doing research. And we've continued to do that, and we've been very successful because I think there still is huge need in training clinicians to be researchers. And this program has been very successful. The current people, for instance, Mercedes being one of them, and Mina being another one, who have been trained as residents and as researchers, and now are part of the faculty. And there are many more. I'm not going to mention all the names because it may not be very useful for you guys, but they are part of our faculty, very successful part of our faculty in bringing in many grants, but also seeing patients, supervising fellows, and supervising residents. So it's really become one of the building blocks of our department, and as such has been very successful, including the training grants that we've received. Mercedes mentioned that the T32, which bridges the period between the residency and research track and getting independent grants. These people have also been very successful. There are a few of them here, and then get their own grants like the K23, R1s, or whatever. So it's been a very successful program. And as a department, because you also obviously want to know how is this department doing? Well, we're doing quite well. We're bringing in, I think, about 50 to 60, well, 50 million in NIH grants, but the total grants is about 70 million. So we're, one of the prominent departments in the country. I think we're currently number 6 in NIH funding, and probably, well, anyway, let's not jinx it. But anyway, we're doing well. We also within the school, we are churning out over 200 grant applications a year. And we're getting funded over a third of them, which is way higher than the NIH funding percentage. So people are doing well. People are enjoying themselves. Fortunately, almost no one leaves us, which is always a good sign. And many people want to come in and come here to work because I think the atmosphere is quite good. We're highly collaborative. We're highly supportive of our faculty from young to old, and trying to stimulate them to continue doing great work. So I think you'll find it a very good environment, conducive to being successful, if I may say so. Both as clinicians, we're able to get the clinicians a pay raise of about 25% last year. So that's good. And then as researchers as well, but people can be very successful in building their careers. Like I said, most of them stay here as faculty, and if they don't stay here, they go to other departments in the country and build their careers over there. Generally as researchers and sometimes as clinicians. So with that, I'm going to leave some time for questions and for Dr. Nestler to provide some information. But again, I think this is one of our great programs in psychiatry in the country, I must say. All right. Thank you.
Absolutely. Thank you, Renee. And we have time for one question for you, and then we go to Dr. Nestler.
Yeah, that always works. You say do you have a
Nothing like asking one question among a group of psychiatrists, because we will interpret you.
Don't feel scared. Okay. All right.
Okay, well, thank you. And now we have Dr. Nestler, who's the director of the Free Membrane Institute, our Dean for Scientific Affairs and Chief Scientific Officer, right?
Yeah. Thanks, Mercedes. So it's really nice to meet all of you. Good afternoon. As Renee said, we think that we have one of the very best psychiatry departments in the country, and especially one of the very best research track residencies. I'll just very quickly give you the highlights of why I think that's the case. As Renee said, our psychiatry department is really one of the elite departments of psychiatry in the country, very broad in its approaches. The psychiatry department is very tightly integrated with all other neuroscience on campus under the auspices of what we call the Free Membrane Institute, or FBI, the Department of Basic Neuroscience, Neurology, Neurosurgery, and other departments are tightly integrated at the clinical, research, and educational levels. And I think we have uniquely at Mount Sinai brought different perspectives and experimental approaches together in the clinical domain as well as in the research domain. We can give you examples of that. Third, we think that we have the formula to make it possible for you all to go from PG1 residents to faculty at Mount Sinai or anywhere else if you choose to go as quickly as possible. So most of the people who pursue basic lines of research can become faculty at the end of their residency, at the start of what would be their PG5 year. Those individuals who pursue clinical research, just because of the nature of clinical research and the increased time that it takes to get findings from human subjects and publish them, perhaps a PG5 year or 6 is required. But again, as quickly as possible on the faculty. We have the funding that makes it possible for you to do that. Mercedes and others in the department will make the maximum amount of time available to you to do your clinical or basic research. That's really important, and we have the flexibility to meet your needs in terms of how you would like to structure that. Some of our past residents have put research strongly up front. Most have deferred it to the later years of residency. Both work. It really depends on a person's individual needs and types of research that you do. And then finally, as you identify a research group to work with, whether it's basic, translational, or clinical, we will make every laboratory available to you at Mount Sinai. You need not work in a faculty member who's a member of the Department of Psychiatry. That could be neurology, neurosurgery, any other basic science department on campus, right? And I think that offers particular opportunities for you all to bridge what expertise you bring to the table as research track residents, as you try to define a unique niche for yourselves in defining your own laboratory, again, to advance to faculty as quickly as possible. Importantly, we only let you work with faculty who understand the unique perspectives of having a research track resident in their labs. We don't want you to do a traditional postdoc, right? Most of you are old enough. You should get a job as quickly as possible. You don't need to do a 5-year postdoc on top of a 4-year psychiatry residency. In my perspectives, I think that's a waste of time, which means that during residency, you should focus your research on your own laboratory, so that at the end of residency, or PG 5-year if needed, you will have built in the data for your first NIH grants, other grant proposals, and establish your independent laboratory as quickly as possible. I think that's the secret ingredient of our success, where you're really not standard postdocs, but you're really faculty apprentices, is the way I like to formulate it, as you move on with your career. I'll stop there and see if any of you are brave enough to ask questions of Renee or me. There's a brave soul. Go ahead, Brendan.
Thank you. I was just wondering, so to my understanding, by PGY4, most of our time would be expected to, I think roughly 75% was the number, would be dedicated towards research. And like you were saying, you're expected to kind of be running your own lab and taking a lot of initiative on your own. I was just wondering, like, some of the specifics of how that would manifest. Would we be recruiting like med students or other residents into our lab? How do PGY4s typically, kind of start setting the stones for having their own lab in research?
Well, most of the research track residents start out by doing much of their own research with their own hands. But depending on the nature of the research, you could start recruiting volunteers even as a PG1. You'll hear from Brian Swice, who just joined the faculty a couple months ago. He finished his PG4 year last June. He was a PG1 resident and recruited a small army of research volunteers the day he started his residency as a PG1. So I think there's many different ways in which you can affect this, and you should definitely ask the recent faculty recruits to the department who followed this path for specific answers to your question. So we should go along. Sahar.
Hi, hello, my name is Sahar, and I wanted to know, you said like we can do our own research, and I want to know, does it mean that there is opportunity for internal funding on top of the the T32 or whatever other funds that are supporting our residency?
Sure. The Freeman Brain Institute does have pilot grant awards that some of our senior residents have successfully competed for. There are some other internal funds that are available through psychiatry and neuroscience. I think that, again, you should ask our recent graduates, but I don't think anyone is short of funds. Our expectation is that our faculty who mentor research track residents will use some of their own funds to get you all started on your career. It's a statement of generosity, right? Because the PI is not going to be owning those data, so to speak, but they'll get part credit for having the experiments done in their lab. And it's a formula that has worked very well.
Thank you so much. That's amazing.
Ethan.
Yeah, hi guys. Thanks so much for doing this.
Oh, go ahead, Mercedes.
I was just going to say, this is the last question.
Yeah.
Cool.
I can tell you're getting nervous.
Yeah, thanks so much, guys, for doing this. I have a question for Dr. Nestler regarding the Friedman Brain Institute. And that is that sometimes with these kind of wraparound institutes that encompass different departments and institutions, that's something that you can read about online, but you can't necessarily feel in person or kind of notice very much. And so I was wondering, can you feel it? And how do you sort of make it kind of operationalize that integration?
Yeah, I mean, that's a great question. I resonate with that a lot. It's actually something I think a lot about as the director. I would suggest, and again, don't trust me, but trust the young residents and recent faculty recruits you speak to. I think the FBI is extremely palpable here at Mount Sinai. Unlike some institutions where the Neuroscience Institute is selective and thinks that there are some faculty who aren't good enough to belong to the institute, that does not happen here, right? So the Friedman Brain Institute encompasses all of psychiatry, all of neurology, all of neurosurgery, all of neuroscience, and many other specialties. We have an annual retreat where we have 600 people attending each year, all Mount Sinai people, not external people. So I think you will if as you get to know us better, unfortunately, these initial visits are always remote, but as you get to know us better, you will definitely feel the vibes of the FBI.
Great, thanks.
Thank you, Dr. Nestler and Khan. And I'm going to share my screen again so that you can see the agenda, and also I can start with a brief presentation of our research training programs. And I will also introduce our, so I'm the Associate Training Director for Research. I co-direct the residency physician scientist research track and the postdoctoral P32 fellowship together with Antonia New. And this year, Brian Swice, who's a graduate of our research track residency, joins us as Assistant Training Director for Research. And Antonia is actually the overall training director for our psychiatry program. So Antonia and Brian, if you can briefly, I mean, Brian's going to speak as a faculty member and recent graduate, so but just give like a 1 minute intro, and then we go to the presentation.
And Mercedes, I'm going to sign off, but I want to just urge you to give all the applicants, Dr. Khan's and my email addresses in case they have any questions. Please feel free to reach out to us. We look forward to seeing many of you here next fall.
Absolutely, same here. Thank you.
Bye.
Bye, thank you. I just want to say hello. I'm Antonia New. I do run the residency program. I'm the vice chair for education, so I also oversee the education of the medical students in psychiatry. I think maybe most importantly, I am myself have had an extensive career in research. And so I think what that means is that I get it in terms of what it takes to become a physician scientist and to be successful in this career. And so I have been very sensitive to integrating the ability to train physician scientists in psychiatry while also protecting a sufficient amount of time for you to be able to build the foundation to be able to be independent investigators on the other side. As Dr. Nestler said, as you finish your training, you've all done a lot of training already. And I just want to make sure that you have the foundation to be able to jump right into it. And my own background enables me to see how important it is that you don't get derailed along the way.
Hi, everyone. And I'll just introduce myself briefly. Hi, everyone. I'm Brian. As some of the folks mentioned already, I was a recent graduate of the program just this past June. And so I transitioned to faculty here as of July 1st. I've been running my lab for the last couple of months, getting everything set up. I know I've met some of you already in the past, and I'll share some of my work in a few moments later in the Zoom. But I just want to make a comment that we all recognize people are coming in at various degrees of how advanced they are versus how much they want to explore other options. And so really tailoring your residency experience and how you could conduct research during residency to the individual is quite a strength here. Some people want to change directions, pivot a bit, explore new areas, go from animal research to human research, or some people want to directly continue the work they were doing prior to coming into residency and to keep the ball rolling. So Mount Sinai is really good at being able to sort of accommodate people where they're at and how to sort of launch them into their independent career. So I'll leave it at that, and I'll share more of some of my work in a little bit. I'm looking forward to meeting some of you folks later too.
Thank you, Brian and Antonia. So I'm gonna share my screen again. So let me pull up the presentation. Okay, can everyone see it? Okay, yeah, excellent. So, so you've met all of us already, but we're listed over here, and we're happy to share all our emails with you. And we've gone already through a brief overview of brain-related research at Mount Sinai, and we've introduced ourselves. I'm now going to be talking a little bit about the structure of our R25 residency research track and our T32 fellowship. I'm also going to briefly introduce a new fellowship co-founded by OMH, which is launched this year. And then we'll have some brief, very brief, we actually had to limit them to 3 minutes this year because we had so many volunteers among our faculty to present, so we couldn't fit all of them. And then, so this will be by faculty and by current trainees, and some of the faculty are graduates of our programs. And then we'll have a final hour between 4 and 5 with virtual networking and Q&A in these breakout rooms. So we'll have current trainees, we'll have our basic and our clinical and translational research faculty, our program directors, and we'll also have empty rooms available for one-on-one meetings. You know, if you strike a conversation with a faculty member or applicant in one of these breakout rooms, you can then leave the room if you want to discuss something in more detail and go to one of those one-on-one breakout rooms. So I'm preaching a little bit to the choir here, but I want to, emphasize why it's important to have physician scientists and clinician scientists in general doing research, and also why I think Mount Sinai is a wonderful choice to do psychiatry residency, or if you're focused in research. Also, if you're not focused in research, we have very strong general programs as well, but also our T32 postdoctoral fellowship. So our Department of Psychiatry has a really robust record of NIH and other funding, as you've heard from our chair, Dr. Khan, and it's consistently, if you look at the past 5 years, it's always in the top 10. This past year, it's ranked number 6 in the country in NIH funding, and our departments of neuroscience and genomics are really, really strong. They're both ranked number 2 this past year, and the Department of Neuroscience and the Department of Genomics are consistently ranked very highly through the years. And there's a lot of collaboration, as Dr. Nestler was mentioning. It's true that there's a lot of cross-pollination and research collaborations across anyone who studies the brain or something related to the brain at Mount Sinai. And this includes multidisciplinary teams. Some are psychiatrists, some are neurologists. There can be others that are interested in cardiovascular disease or immunology, you name it. And there's this very focused institutional effort to really support clinician scientists and translational research, and to understand what it takes to hire and retain, or train, hire and retain physician scientists and clinician scientists. I'm not going to talk a lot about this, but I have a slide just showing we're really in an extremely diverse neighborhood. New York City is a very diverse city, and we also have a lot of institutional efforts towards increasing diversity, equity, and inclusion at Mount Sinai. For those of you who may be interested, you can mentor, for example, trainees and students in some of our pipeline programs to enhance diversity. And this is something that our T32 fellows and residents have done in previous years. And again, preaching to the choir here, so I'm not gonna spend a lot of time in explaining why we need clinician scientists in psychiatry. I'd rather just go to the structure of our tracks. So the first I'm going to talk about is our physician scientists research track in psychiatry. So for those of you who are applying for residency this round, this has a separate match number. This is the match number that you need to apply for. And this one is really designed for applicants who have very substantial research experience. And we often get the question, is this limited to MD, PhD, or MSCP graduates? The answer is not, but you have to understand it's very competitive, and priority is usually given to those who have this substantial research experience, and that often means having an MD and a PhD, but we have also other residents and prior graduates that had an MD and some other form of substantial research experience, like a postdoc, or they had an MPH or some other experience that provided that research background. These are some of the advantages of the R25 funded physician scientist research track. So this is a very strong program. It was first internally funded, then funded internally and through the Leon Levy Foundation. It's been running since 2008. I'm actually one of the first graduates, together with Dr. Panos Roussos, who's also an extremely accomplished researcher and clinician scientist. And we really know how to do this. Antonia and I have been co-leading this program for, I guess it's now 7 or 8 years, I forget. And we now have R25 funding as well, on top of all the internal resources. So in terms of the financial advantages, you are eligible like any other resident to get benefits, housing. In addition, you get some tuition funds that you can use to take any courses and pay for the credits. You also have pilot research funding. It's $27,000 per resident across the 4 years, so divided. The total amount is $27K. $3,000 per year in travel funds to present at conferences. And then you're eligible starting usually in the 2nd year to apply for the NIH loan repayment program, which can give you up to $50,000 per year as long as you're engaging in qualified research for an average of at least 20 hours per week. The structure of the track, and I'm going to show this in a figure form, is such that you get increasing amounts of research. And again, this is the standard template, but this can be tailored to the individual needs of the resident. In cases where it really makes sense, and Ryan Schleiss, for example, was one of the cases where it made sense to change this structure, he actually had protected research time even in the 1st year, because he had these projects that were really shovel ready and needed that time to be front loaded. But this is the standard that we figured out works for the majority of research track residents, which is that the protected research time really starts in the 2nd year. But then once it starts with a protected research block of full-time research, so that you can embed yourself in your mentor's lab and develop those collaborations that you need to for your growth as a researcher, then it continues longitudinally. So you see about a third, and this would be like 1.5 days during your second half of the 2nd year, and then 50% in the 3rd year, and then 75%, which I think is really even higher than 75, in the PGY4 year. And this is an example, you can see this on the website, of what is expected in each of the years. But again, this is tailored through an individual development plan to each individual trainee to make sure that we meet you where you are in terms of background and readiness to launch your independent career. And the second program that I'm going to briefly present is our T32 postdoctoral fellowship, and this is geared towards training clinician scientists. This is also, if you're a trainee, you're eligible to apply for the NIH loan repayment program, and this provides funds for tuition, for travel, and you're also eligible for benefits including health insurance and also housing and childcare stipends. And this one is open not just to MDs and MD PhDs, but also to any other mental health related doctorate degree holder. For example, if you have a doctorate in social work or in nursing, we have many psychologists, PhDs, PsyD. And this is just to show about, we really want to retain you, and Dr. Khan has mentioned a little bit of that, as well as Dr. Nestler. When we think about recruiting for our research tracks, we really think about who's going to be faculty in 5 or 6 years. And we've actually been able to retain or recruit back over 50% of our clinician scientist graduates from all of our programs. And in fact, for residency, for example, this year we recruited 100% as faculty, right? So this is actually a very common occurrence. And then our graduates have also been extremely successful in obtaining independent funding, some of them immediately after graduation. I want to also remind everyone, besides the T32, we also have other opportunities for postdoctoral fellowships if you need additional time after residency, including some of them for which visa holders, non-citizens, non-green card holders are eligible. And now I just have I'm over time actually, but I do want to present. There's this new fellowship that is a partnership between Mount Sinai and the New York State Office of Mental Health, OMH. And this is a public psychiatry research fellowship. If anyone is interested in this fellowship, the goal is to train researchers to be investigators focused on eliminating mental health disparities, and it's 80% protected research time, 2-year fellowship. Psychiatrists are eligible after completing a PGY 4-year of residency. And Dr. Sydney Hankerson, who's our Vice Chair of Community Engagement and the Director of Mental Health Equity Research, is the Fellowship Director. So if any of you is interested, this is his email, which I'm going to share later as well. So now I want to give the...
Mercedes, just one quick. So anyone interested in services research, he's really an expert in services research.
Absolutely. And I'm going to now let Nina be the host for the brief faculty and trainee presentations. And this is the list. And I'm going to end the show so that we can see everyone's presentations as well. So let me stop sharing. Yep.
Thank you, Dr. Perez. I think we're going to start with Dr. Hurd, which is here.
Thank you. Let me just quickly share. Actually, I am just running between, well, I ran out of our clinical team meeting. So we have, we conduct clinical trials here at the Addiction Institute. I'm the director of the Addiction Institute at Sinai, just for some of you who may not know. And we have a pretty huge clinical population of individuals who suffer from substance use disorders. And so we do many different types of clinical research, whether it's, you know, ideas that we have based on our science in terms of investigator-initiated projects. We have company-sponsored projects, and also we're part of the National Clinical Trials Network that the National Institute of Drug Abuse oversees. So we're interested in the biological underpinnings and really developing new medications that are not addictive for individuals suffering from a substance use disorder. And this is actually based on a lot of my team. We do both preclinical and clinical studies. One big project that we're working on right now actually started with our preclinical studies of cannabidiol, as we study the effects of cannabis in relation to addiction and psychiatric risk. But we also saw that in contrast to THC, CBD actually reduced heroin-seeking behavior in our rat model that we translated to human studies. And we've now passed phase 1 and actually just finishing phase 2, and just recently got the approval from NIH to now move to the phase 3 in studying the effects of cannabidiol in opioid use disorder. It'll be about 300 people who will be studying. So we are happy to get help in this work. In addition, we also do neuroimaging of CBD in trying to understand the neurobiological underpinnings of why it may be impacting on craving and anxiety and related symptomatology in opioid use disorder. We also, as I said, are part of the Clinical Trials Network, and Dr. Karen Baki is a site PI for a number of those studies where we're looking at novel treatments that are not yet FDA approved for example, psychostimulant use disorders, because there's no FDA treatment for stimulant use disorders. So we're part of that. And those are some things that I think that, again, if, looking at buprenorphine, naltrexone, and extended release buprenorphine for cocaine use disorder, but we're also looking at the NIH wanted us to also look at ketamine as a potential treatment for methamphetamine use disorder. And those are also 2 studies. We have other studies that are company sponsored in tracking, for example, well, I know this one is a grant for Welcome Leap in looking at epigenetic biomarkers in the blood in PBMCs of individuals with opiate use disorders, because we saw again from our animal models that we were able to track certain epigenetic changes happening in the brain are also happening in the periphery. And if we can find biomarkers for substance use disorders, that would be great. So it has hundreds and hundreds of people who are, the data will be collected, and we already have the PBMCs and we'll be analyzing those. And there are data that we have already from different studies. Again, people can contact Dr. Karen Baki for one of the projects that she had from mentalizing deficits in cocaine use disorders, looking at immune and neuroimaging data. And a lot of that data is there as well to some of our epigenetic stuff. And, you know, for us, it's for those of you who may be interested in the preclinical models, and also we study the postmortem brains of individuals with substance use disorder and do a lot of sequencing and looking into the relationship to as neurobiological targets for developing new medications that then transition back to our clinical studies. So this is just to give you an overview of, you know, the translational approach that we have. And so, like I said, we cover all preclinical to actual phase 3 and even phase 4 clinical trials. So please reach out to me if you have any interest or questions.
Thank you, Dr. Hurd. Thank you. We're going to move on to Dr. Denise Kai.
And so I'm associate professor in neuroscience. I'm also the co-director of the Computational Systems Neuroscience Center within the Friedman Brain Institute. So my lab is really interested in how is it that our brain is able to accumulate what seems like an infinite number of memories across a lifetime, and how do we store that in our finite, brain space. I specifically study mice, and so one of the things my lab does is develop neural technology. So we develop these miniature microscopes, like the size of this part of my thumb. You can put it on the animal's head, and while the animals are exploring the world and sleeping and interacting with other animals, or whatever you want the animals to freely do, we can record hundreds to thousands of cells and their neural activity in vivo. And so we can track these same cells in these animals across 2 to 3 months, which for a mouse is quite a long time. So while my lab is really interested in memory and memory encoding and storage and retrieval, and how to separate memories, forget memories that you should not be storing. This has a lot of implications for as we think about trauma, and we have looking at how trauma forever changes how animals learn in the future. We're also interested in how this goes awry with aging and Alzheimer's and other neurodegenerative diseases. So what would you get from being my lab, right? So maybe you don't want to study memory per se. So Brian Swice, I was at the pleasure of co-mentoring Brian, and he does decision making. So he got access to all these tools that we're developing. So he gets firsthand access before they're published, collect preliminary data, and getting access to brand new technology to ask questions you couldn't previously ask helps with the high risk, high reward grants and other grants, but also with answering scientific questions. Anyways, if anyone's interested in chatting with me, I will be at the, I guess, a later breakout, or feel free to email me. Thanks, everyone.
Thanks so much. Dr. Cheryl Corcoran.
Hi, so I'm Dr. Corcoran. It's nice to meet you all. I originally did my training at Harvard College Medical School residency. I was then at Yale, and then I was at Columbia for a while. And I just want to say that my career has really taken off since I came to Mount Sinai in 2017. So I want to echo what Dr. Khan said earlier. People come here, they don't leave. I couldn't imagine leaving. And I've really had the resources, intellectual and material to do my work. I am a schizophrenia researcher. I have been for a while. When I was at Columbia, I was there for 16 years. I started a clinical high-risk program because we wanted to identify people who, young people who seemed like they were at real risk of developing schizophrenia. It's been a great field. I've started a clinical high-risk program here, and Dr. Khan and I and others are part of this huge consortium called AMP Schizophrenia, which is international, 32 sites, really collecting all kinds of data to try to predict who will develop schizophrenia. In that consortium, my area of expertise has been computational phenotyping. So around the time that I came here in 2017, I was really grappling with how in neuroscience, Columbia, the neuroscience like here is beautiful, how it was quantified, what we learned about the brain, and at the same time, how primitive we were compared to other fields in medicine in terms of quantifying, operationalizing the main variables that matter to us in psychiatry. And what that is language and it's behavior. And it's how people behave in the context of being with other people. And all that we have really had for that has been clinical rating scales. And it's just been great working with Dr. Khan because he agreed with me. And so we've been off to the races. So let me just show you, I don't know how much time I have, but just briefly, some of the work that I have done with Dr. Khan and others.
I have 30 more seconds.
I have 50 seconds. All right, we've used computers to characterize language and face expression. Language is a window into the mind. Steven Pinker, MIT. Long before there was ChatGPT, I was collaborating with people at IBM, computer scientists, engineers to use generative large language models. We modeled coherence, the people being tangential in schizophrenia. We've moved on now to look at face action units. People with schizophrenia have flat face affect. This work comes out of Darwin. This is all that you need in terms of materials. And these are the time series analyses that we're looking at in DIADS to look at behavior in a quantitative way in psychiatry, which is desperately needed. So I've done this in schizophrenia. My colleagues who do autism, Alzheimer's, depression, they're adapting these same things. Dr. Khan and I have this very large U01 predictor grant, it's called. It was advertised by the White House. We're going into the clinics and we're going to record clinical sessions, clinicians and patients, so we could really try to operationalize what are pathological features that like may be related to suicide risk, et cetera, but also looking at the therapeutic alliance. And in addition, this is the last thing I'm going to say, we now have a new grant where we're going to build a lab here at Mount Sinai and look at DIADS and do physiological recording. So we could also look at eye contact, we could look at heart rate, and we could do hyperscanning with EEG and fMRI. Very interesting work. Please join me. Happy to help you soar. That's it. Thanks.
Thank you so much. Now moving on
Let me
to
stop sharing.
yeah. Moving on to Dr. Roman Durand from Scott Russo's lab.
Yes, can you hear me?
Yeah, we hear you.
Okay, I'm gonna share my screen. All right, can you guys see that?
Yeah.
Okay. This is Roman Durand. I'm speaking on behalf of Scott Russo, that unfortunately couldn't be here today. In the lab, we are mostly studying how brain and body interact in the context of model for psychiatric disease. And our work is at the intersection between two big fields, neuroimmunology and circuit neuroscience. So we're using techniques such as flow cytometry, omics, transcriptomics, proteomics to investigate neuroimmune interaction in the brain and in the periphery, and circuit neuroscience to look at the activity of neurons, for example. As Dr. Kai mentioned, we're using calcium imaging to probe for neuronal activity, or using some techniques to manipulate the activity of neurons, such as chemogenetics or optogenetics. And we used animal models of psychiatric disease, mostly stress models. So we have social stress or early life stress. And we then probe for behavior in animals mostly. And we're also trying to build new behavioral models that we deem translational, because we try to reverse translate them from the clinic. I think we're trying to build a bridge between the clinic and animal research. And also to mention, we receive a lot of samples from our collaboration from the clinical department at Mount Sinai, especially our collaboration with the laboratory of Dr. James Morrow. And we have this very ongoing continued effort to try to go back and forth between the clinic and animal models to probe for neuroimmune or circuit alterations. And just as a real quick few example of the work we're doing, recently we've been using tissue clearing to look for whole brain activation following exposure to social stress. And from there, we were able to identify circuits that were critical in the emergence of social deficits following exposure to a social trauma. So that's one of the technique that we're currently using in the lab. Using this technique again, we were able to probe for whole brain networks playing a role in the emergence of aggression in males and female mice, and using techniques coming from the transcriptoming world and bioinformatic analysis, we were able to identify networks of brain region that plays a role in the emergence of this aggressive behavior. And then using state-of-the-art circuit technique, we're able to manipulate these brain regions to try to manipulate the behavior. And as a last example, we're trying to build the bridge between the periphery and the brain. And for that, for example, we used whole brain clearing to look at the infiltration of circulating monocytes in the brain parenchyma and to try to identify the impact of these circulating immune cells in changing the activity of the brain and ultimately the behavior. And when we have the opportunity, we try to validate these findings in our clinical population here at Mount Sinai. All right.
Thank you so much, Roman. Moving on to Dr. James Morrow.
Hey, everybody. Hey, Roman, that was great and a great segue. It was fun. Our group got a chance to be part of that work. So nice to see everybody. I'm James Morrow, psychiatrist, professor of psychiatry and neuroscience here at Mount Sinai. Hey, Ethan, good to see you here. Hey, everybody, I recognize some folks. So I direct the Depression and Anxiety Center for Research and Discovery here. And we're primarily a clinical and translational research group, very much patient-facing. And what we like to do is interface with folks like Rahman, who you just heard from, great stuff going on in neuroscience, to get ideas about how to test and translate novel insights in mood and anxiety spectrum disorders, stress-related disorders from rodent models in humans. Maybe I'll give you a couple examples of some of the projects we're doing now. Apologize, I don't have any slides, but so for example, one of the projects that we're just wrapping up is, those of you might be familiar, the National Institute of Mental Health has a specific mechanism for that they call experimental medicine to identify new treatments, novel targets. So we got a signal from our basic science colleagues for a specific family of potassium channels called KCNQ channels that were identified from stress models in rodents. And then we found a drug that we could repurpose that was marketed for a different indication that was a positive allosteric modulator of that target. We got a grant to test whether that drug would actually affect brain circuits using functional MRI in patients with depression in a way that, again, we sort of designed the clinical trial to mirror what the endpoints they were seeing in rodents. And we're actually in our 8th year, if you can believe it, of a large staged grant. We're about to finish our 2nd randomized controlled trial. That will be the 2nd randomized controlled trial ever of this target in depression. So very excited. We've partnered with a drug company that's gotten interested in this to study a next generation KCNQ opener. They got so interested, they started their own this is a target that was initially being developed for seizures. This is a neurology company, and they're actually developing their own program to test it in depression as their first foray into psychiatric illness. And really, if you talk to leadership at the NIMH, for those of us that are sort of in this space of clinical research, translational, what they want us to do is they want us to take federal dollars to de-risk new targets from basic science to lure in industry partners to actually invest in developing agents that could be moved through regulatory approval in the US and elsewhere for novel treatments for mental illness. So very exciting. That's kind of what we do. Other stuff, very interested in immune. And as Roman knows, and we were very happy to be part of that Nature paper that came out a couple of months ago, super exciting. And we're working on some novel targets that was developed from that area as well. So that's the kind of work we do. I think I'm probably at time, so I'll stop talking now. Thanks, everybody.
Thank you, Dr. Morrow.
Thank you.
Moving on to Dr. Lauren Lipau.
Great, thank you. I have a few quick slides I'll share. All right. Okay. Hi, everyone. I'm Lauren Lipo. I recently graduated from a former track at Mount Sinai, the psychiatry the psychiatry residency plus neuroscience PhD research track. That was a 7-year program. And I just transitioned to assistant professor on the mentored research track. So I came in with a lot, maybe less research experience than you all, and had a lot of time during my 7 years to gain experience. And now I'm on the mentor track, and I'm also a T32 fellow this year. So happy to talk to you about this transition from training to assistant professor. So I'm going to be telling you about the Center for Psychedelic Therapy Research, which is both at Mount Sinai and the Bronx VA. So I think what makes our psychedelic center really special and unique is that it was born out of the lab of Dr. Rachel Yehuda, one of my mentors, and she studied the biological mechanism of action of change in treatments for post-traumatic stress disorder and trauma-related disorders. So our lab is really focused on collecting biomarkers and also consists of clinicians such as clinical psychologists, psychiatrists at the VA in the PTSD program. So when we design our trials, it's really with the participant in mind from clinicians who are so experienced in treating PTSD. So for our studies, we collect a lot of biomarker data ranging from hormones, genetic, epigenetic, neuroimaging, and now audiovisual data as well, along with, of course, the clinical data. And so as part of this center, I've gotten to participate in some really cool research. I can't tell you about all of our trials, but I will tell you about a few. So we completed an industry-sponsored psilocybin-assisted therapy for PTSD study. So it was really eye-opening, I think, to get to be an investigator and a study therapist on this study, where I got to not only work with this new treatment, but also really understand what it's like to work with industry. And then I'm a lead on a study called Processing Intergenerational Trauma with Psilocybin-Assisted Therapy. And I'm currently taking it through the intense regulatory process through an IND at the FDA and just getting a lot of experience there and some independence. So that study will be up and running by the time you would reach our residency. And then something that's great that so many people here have mentioned is that at Mount Sinai, we're super collaborative. So we have several collaborations, but some of which are with James Morrow, who you just heard from, on a clinical trial studying the mechanism of action of psilocybin, and then also the first randomized controlled trial comparing IV ketamine to ketamine-assisted psychotherapy. And again, I've gotten to participate in kind of all aspects of those studies as a fellow and trainee, and now also as an assistant professor. So at the VA, we also just completed, and the data was just locked last week for a 2 versus 3 doses of MDMA plus therapy for veterans with PTSD. And so I'm so excited to get my hands on this data. The results were incredible. We haven't published yet, so I can't talk too much, but we have so much biomarker data, and we have a basic science kind of wing of a wet lab as part of our center. So I'm so excited to kind of dig into some of this data. And if you were to join you'd be welcome to collaborate with us and to ask your own research questions too, if you're interested. And then my particular area of interest is in something I noticed during residency training, being a clinician and scientist, is really this gap that I know Dr. Corcoran was describing earlier, of some of our neurobiology becoming so sophisticated, yet feeling kind of more and more distant from the clinical experience of sitting with the patient. And especially because I think at Mount Sinai, you get a ton of psychotherapy training. You develop this, I think, wisdom and intuition that can feel really far from neurobiology. So my interest is in, this is me as a fake and some fake participants developing my clinical expertise. And then this is analyzing some audio data. So I'm starting to collect my own pilot data for my career awards, collecting and analyzing AV data of both the transcripts and also the video clips of psychotherapy in our psychedelic trials to better connect our biomarkers to kind of the human experience of healing from trauma. So this is also in collaboration with my mentor, Alexander Charney at the Charles Bronfman Institute for Personalized Medicine. As you can tell, there's so many institutes at Mount Sinai. So really whatever you're interested in, someone around Mount Sinai is doing it, and there's always an open door, especially for clinicians. So if you're interested, please contact me. This is my email address, and I hope to speak with you in the breakout rooms. Thanks.
Thank you, Lauren. We move on to Dr. Brian Swice.
Hi, everyone. I'll share my screen as well. All right, so I just wanted to give you all just a brief taste of the kinds of approaches that I use in my sort of research. So I primarily do animal research. I've been studying how rats and mice make decisions, studying behavior with a bit of translational relevance to humans. So I kind of have this cartoon schematic here just to give you a sense of the types of questions we ask. So in this cartoon, you see a rodent at a fork in the road deciding to turn left or right. It could be for a food reward. It could be for a decision to interact with another mouse. But the core of my questions that I've been tackling for over the last 10 years have been asking, how does an individual deliberate over their choices that they might be choosing between? How do they imagine the future when deciding what one should or shouldn't do? And how does the brain also access past experiences or even recent decisions? And how does that inform future choices when reflecting on what one did? So in this cartoon example, you can see this cloud represents a thought that an individual might be having, thinking back to a choice the animal may have made on a previous trial. In this case, maybe the animal walked away from a food reward, in which case it maybe could have decided to accept this reward instead. And how is that prior choice experience being accessed and represented while an animal is deciding what they should do in the current moment? Maybe the individual might be thinking about the reward they missed out on, maybe the action they shouldn't have done or the action they could have taken instead. And so these sorts of questions sort of underscore the higher order types of concepts in cognition that I'm interested in studying. Not only how does the brain represent what they did, but how does the brain represent what one could have done instead? And so this underlies a lot of complex higher order forms of cognition. And this also underlies aspects of different emotions that individuals may process, like how does an individual learn from what they could have done differently? And how does this underlie the basis of regret and these other complex emotions that we're trying to tackle both on the animal and the human front? So some of the approaches we use, Denise had mentioned, one of my mentors while I was resident, this technique of using mini scopes where you can implant a miniature microscope into the brain of a mouse. These microscopes are about the size of a nickel, and you can have an animal run around a maze performing various tasks while you're also simultaneously recording from and imaging the brain. And so here's an example of what this camera implanted into the brain can image. Here you're seeing individual neurons that are firing, and so we can record activity of these cells over long time scales and large populations of neurons. And we can ask questions like, how does the brain represent different aspects of a choice, or in this case, different areas of a maze while an animal's thinking where it is currently versus where it might go next? And we can see this in real time as individuals are sort of integrating both past events and future outcomes while they perform this task. Ramon also mentioned some approaches like tissue clearing, which I've collaborated with him quite a bit during my time in residency, where you can take the entire brain and do whole brain imaging to gain access to various biomarkers that might be relevant for your task or disease model of interest. So this is kind of some of the tools that I've been learning how to implement in my own research over the course of residency. And now that I've opened my lab a couple months ago, we're integrating these sorts of tools to ask questions just about how the brain makes decisions and how decisions can become dysfunctional in a whole host of different disease models, and how we can translate some of these findings into the clinic with patients. So I'll stop there.
Thank you, Brian. Now we're gonna move on to Deepak.
Thanks, Mina. So my name is Deepak Kaji. I'm a PGY3 research track resident at Sinai. I work with Panos Roussos in the Department of Genetics and Psychiatry. My research employs stem cell models as a tool for studying neuropsychiatric disease. And today I'll be focusing on Alzheimer's. So in Alzheimer's disease, we have this protein called tau, which can aggregate into neurofibrillary tangles. And in Alzheimer's disease, these tangles accumulate throughout the brain, but they start off in these very visceral parts of the brain, like the hippocampus and the entorhinal cortex, before working their way a little more superficially into the limbic system, before finally reaching the most superficial part of the brain in the neocortex. And we know that cognitive decline gets serious when tau finally penetrates the neocortex. Interestingly, there's a disease you've probably never heard of called PART, primary age-related tauopathy. And in PART, tau accumulates in those deep regions of the brain that it does in the Alzheimer's disease, like hippocampus and the entorhinal cortex. But it never gets past the limbic system. In PART, patients experience mild to no cognitive impairment, and the vast majority of people get PART. And what it basically tells us, the big difference between PART and Alzheimer's disease, where people get really sick, is whether or not that tau enters the neocortex. And it suggests that tau abundance matters, yes, but more than location, more than abundance, it's location that matters for understanding cognitive decline. That's the big thing. So how can we go about using stem cells to study neuropsychiatric disease? Neuropsychiatry and Alzheimer's demand living human models of disease. And pluripotent stem cells come from human patients' blood and fibroblasts, and they get reprogrammed. And once they've been reprogrammed, they have the capacity to become any tissue in the adult body. Using differentiation protocols, we can force our stem cells to represent these things called organoids. These are three-dimensional living models of human disease. And these three-dimensional organoids will recapitulate key features of distinct brain regions, like the neocortex or the hippocampus. And when I load the genome of the pluripotent stem cells with an allele like ApoE4, which is the most common sporadic driver for Alzheimer's, the organoid will now recapitulate key features of Alzheimer's disease, such as elevated levels of beta amyloid plaque and elevated phospho-tau. So how do I go about using these things to study Alzheimer's disease? Well, I can certainly look at these organoids independently, looking at how regional differences in the brain influence tau accumulation. But since tau migration matters so much, let's say I wanted to study that. Well, I can take my organoids and I can fuse them together. And I can manipulate the genotypes of these organoids independently, such that the hippocampal half could produce tau, but the neocortical half can't. And then I can fuse them, and I can leave them fused for a few months at a time. And then months later, I could use a scalpel to cut them down the middle and re-separate them, looking at each half independently and how well that tau migrated and under what conditions it migrated. Then I could look at that with cheap technologies where I got tons of information, things like ELISAs and immunohistochemistry, where I can make tons of slides, or I can make it a little bit more interesting. I can use something like spatial RNA sequencing to understand how tau is disrupting the transcriptome along the neocortical hippocampal axis as it migrates. All in all, I think stem cell biology is giving us like a remarkably powerful tool to do mechanistic work in three-dimensional living human models of disease. I'm really excited about being able to use stem cells to study neuropsychiatric disease, and I thank my lab and Sinai Psychiatry for giving me the chance to talk today. If you have any questions, please reach out.
Thank you, Deepak. It's only me left, so I'm just gonna try to be as quick as possible so we can move on to the breakout rooms. Do you see my slides?
Yeah.
Okay. Okay. So my name is Mina. I do clinical research, and I want to present a little differently. I do clinical research, not fancy stuff as Brian, Deepak, and others. Uh, but I want to show you briefly how the robust supportive system of Sinai physician scientist track has helped push my research forward during residency. So before residency, I was a postdoc at Columbia. I did some research on the neurobiology of suicide, had some experience in neuroimaging and clinical trials. Around this time of the application season, I reached out to potential mentors in the place I applied to. Dr. James Morrow graciously accepted to mentor me at Sinai. In my PGY 1 year, there is no official dedicated time for research. I didn't do much during the first 6 months as I was mostly off service. But toward the end of the year, I started bi-weekly meetings with my mentor to explore potential projects. I also hired a research assistant to help me with a NARSED grant that I had received before residency, submitted a review I had been working on, and I attended the APA Research Colloquium for Young Investigators. PGY2, I had 2 research days per week during the first half of the year. So I split my time between working on my NARSED study and seeing patients enrolled in clinical trials at the Depression and Anxiety Center led by Dr. Morrow. Later in the year, I had 2 full-time research blocks, so that's 2 months. So I had a chance to submit like a couple posters at SOBB. Also got some regulatory approvals for a new clinical trial at the Depression and Anxiety Center. And then in the last 4 months, I returned to clinical service, so I had limited time for research. There comes PGY3 at 50% research time, or about 20 hours per week, with more flexibility, given the nature of the outpatient work. So I continued seeing clinical trial participants, submitted another poster at ACNP, went to a few conferences thanks to the R25 funds and some travel awards. And then I had a chance to run some secondary analysis of data provided by my mentor to use as a pilot data for my K Award application. In addition, I submitted like a few grants to the American Foundation for Suicide Prevention, Lynn Levy Foundation, other places, but I didn't get any. Now in PGY4, I have over 80% dedicated research time. Plan to submit my K23 application in October. I'm also resubmitting all the grants that I didn't get last year. Hopefully one of them will come through by the time I graduate. My work includes multiple projects that I'm really excited about. My K23 application focuses on in vivo imaging of blood-brain barrier integrity and its relation to peripheral inflammation in depressed individuals at risk for suicide. I'm also planning to study the immune mechanisms of anti-suicidal effects of ECT and ketamine if that gets funded. Currently working on writing up the results of my NARSED study on the spatial and temporal characteristics of buprenorphine prescription rates in relation to suicide and opioid overdoses. Helped Dr. Morrow with a grant, R61, R33 grant, studying a monoclonal antibody for treatment-resistant depression. We also collaborate with folks at Columbia on another similar study. I also help out with other clinical studies at the Depression and Anxiety Center, including those on dietary polyphenols, tynaptin, ketamine-assisted psychotherapy, psilocybin, and others. Thank you.
Thank you, Mina. So now, and thank you all for being mindful of time in the presentations. I know we had very little time to present each lab. So now I'm going to share my screen again so you can see all the breakout rooms. 1 second. Okay, so there's going to be 1 breakout room for program directors, 1 for current trainees, basic and clinical and translational research faculty, and then there are rooms available for one-on-one meetings. What I'm going to ask, so that we make things easy, is that those who are students or trainees, applicants, potential applicants to the tracks, that you identify yourself with your name, training level, and the school. And then if you're faculty, if you could like make sure that in your Zoom name, there's something identifying you as faculty. And then we're gonna go to the breakout rooms. If you have any questions, you can reach out to Hetal Patel, one of our program coordinators, on the Zoom chat if you have any issues moving from room to room, but you should be able to self-assign yourself to one of the breakout rooms. So I'm gonna end the show and stop sharing so that we can all go to the breakout rooms, and you should see them all already open. And I'm gonna go to the program directors one, so I'll see some of you there. And thank you all for having joined our virtual meet and greet this year. And this is being recorded, and it will also be available on our website. So, and 5 minutes before 5 p.m., we'll all go back to the main room so that we can wrap up the session. Bye, everyone. See you later. Hi, everyone. Yeah, we're back here just to say thank you all of those of you who have participated as faculty, those who have presented, and also thank you to all of our applicants. And feel free to send us an email if you're interested in hearing more about the program. I'm going to put my email on the chat. And this is recorded. This is going to be on the website. I encourage you to be in touch, if you have any questions. And, this has been a great session. So thank you to everyone.
Wait, Mina, you have there's a typo in your email.
Oh.
Oh.
Yes.
Oh, right. Well, thank you for.
And best of luck for
best of luck. It's a pleasure to see everybody here. We're eager to meet all of the applicants, and thank you for the faculty for coming, and for the physician scientist trainees too. Thank you.
Absolutely. Thank you, everyone, and clinician scientists. So we have some psychologists and
too. Yes, I'm just seeing. I'm looking all around, seeing who's who. Okay.
Okay.
Thank you, Mercedes and Antonia, and also
and Mina. Thank you. You organized a lot. Mina's the star organizer.
Mina, you did a great job.
Thank you.
Thank you.
Okay. Thank you.
Bye. Have a nice afternoon.
Here in New York.
Bye.
Bye.